Today, I'm very excited to briefly introduce my group studying the ecology and evolution of host microbial interactions. Okay, so what I think is a big open question in microbiome science is to identify key host microbial interactions that affect health in complex systems. And what I mean by complex system is like the vertebrate gut microbiome. So in relatively simple systems like insects and their symbionts, these key host microbial interactions are pretty well established. So when you remove the symbiotic partner, it can significantly affect the host. And this is a result of a process called coevolution, which is a history of reciprocal selection between the partners. And this evidence of coevolution is limited in the vertebrate gut, in part because there's hundreds and thousands of microbial species. But if we can find these evolutionarily stable associations, these can be a therapeutic target. But how can we look for these evolutionarily stable associations? So one way is to look for patterns. So patterns of shared evolutionary history, which creates the opportunities for coevolution. And another way is to look for mechanisms. So say vertical transmission or host genetics can generate this pattern. So I'll touch on these topics today. So to look for the shared evolutionary history, we can create the host phylogeny and the bacterial phylogeny. And when we see this matching pattern, this is the pattern of co-diversification. And on the extreme end, when you see this mismatch pattern, it suggests these strains are being transferred between these host populations throughout the history. So the idea is to try to rank, say, the human gut microbial species by their degree of co-diversification. And that's what I did during my postdoc, genotyping the human genomes and the microbiome from 6 countries. So here I'm showing you the host phylogeny, the human phylogeny. On the right, these are bacterial phylogenies, the ones that share the history and the ones that don't. So the ones that look pretty similar, these co-diversified microbes, it suggests that these microbial strain has been transmitted across generations since out of Africa for thousands of generations. So these are great candidates to study further. So now let's think about the mechanisms that can generate this pattern of co-diversification. So one is vertical transmission. So we can identify microbial species that specialize in vertical transmission. And this is what I did during my PhD using wild-derived inbred lines. So when you plot the transmission score on the x-axis, it shows it's not a normal distribution. It shows a significant bimodal distribution. So this suggests that there's some microbes that are specializing in horizontal transmission, and some are specializing in vertical transmission. So the ones that transmit vertically overlap with these list of species called co-diversification as well. And another mechanism that can maintain these stable association is host genetics. So we're also interested in identifying host genomic regions that can predict these microbial variation. So we use approaches like microbiome genome-wide association studies. So I've done MGWAS on wild mouse populations, identifying genes that can predict abundance of bacteria. And similar studies have done in humans. I'm listing some examples here. But when you compare the candidate gene drift from humans and mice, there's a significant overlap between the two, more than you expect by chance, suggesting that there can be common genetic mechanisms across mammals to maintain these microbiome variation. So that's from the host perspective. But from the microbial perspective, new PhD student Caitlin Sosi, co-advised by Rosie and I. So she's leading this project to identify these members of the gut microbiome that's controlled by host genetics. So we're using these wild-derived inbred lines collected from these locations, and you do an embryo transplant for existing mouse line, so we can expose these different genotype of mice to the same mother, same microbial pool, so we can study which component of the microbiome is controlled by genetics. And perhaps more clinically relevant, more interesting questions for this audience might be to study the health consequences of disrupting these stable associations. So here's an example of co-diversified microbes, and these arrows are pointing to this evolutionary mismatch. So for example, here, there's an African individual carrying a European strain. So the question is, what's the health consequence of this mismatch? And at least in pathogens, we know it's been reported this evolution mismatch is linked to different pathology. So Helicobacter pylori is a famous example of following our migration history, and it's also a causative agent of gastric cancer. So in this case, if you're an African individual having an African strain, pathology is mild. If you're non-African, the exact same African strain, your pathology can be severe. So it's very interesting. And besides these pathogens, we're interested in identifying commensal gut microbes that we can study this evolution mismatch and the health consequence. So we're planning to do this in two parallel systems, one in immigrant populations in Phoenix, and another is to use wild rodent population in the Arizona Sky Islands. So we think immigrants are carrying their native strains from their home country to Phoenix, but how long these strains persist, right? And what happens if these strains get replaced by foreign strain or gets lost, right? So we're interested in the health consequence. And we can ask similar questions in this parallel system in wild rodents because these animals are isolated on top of the mountains about the same time as the human population divergence. So these can be a great model to do experiments in the lab, say exposing them to foreign microbes. So at the end, we can identify host phenotypes that can be predicted by this evolutionary mismatch or look for traits that's affected by native and foreign microbiome. So in long term, we're interested in studying the extent of coevolution in mammals and studying their implications in human health. So thank you for listening. And I don't think we have time for questions, so please come talk to me later. Thank you.